we study single molecule biology for a simple reason, because biological processes are highly complex and high dynamic. And if we can film the molecules one by one, we can learn a lot about the mechanistic details and how nature works. What you see in this movie is a graphical representation about how DNA replication works. And as you can see, it's a combination and a coordinated action of many, many individual enzymes. So when we're looking at processes like that from a bulk perspective, of course, we miss a lot of this detail. And so it would be great if you are having a technique that allows us to really zoom on these individual molecules and observe what is happening in real time to each and every single protein. And this is the reason why we developed the C-Trap, which is our flagship instrument that combines optical tweezers, single molecule fluorescence microscopy, and microfluidics. And this really enables the filming of molecular movies. As you can see here on examples where you see two glass spheres which are trapped by optical tweezers. There's a single DNA molecule suspended in between, and what you see moving very fast in a random fashion along a single DNA molecule, those are individual fluorescent proteins. There is a large team also behind this mission of unlocking single molecule methods, which is composed of young and energetic scientists and engineers from a diverse background from all around the world. So, but let's first start maybe a step back. So what are optical tweezers? Optical tweezer is essentially a technology that uses light focused by an objective to really physically trap a small microsphere in the light focus. Once we can trap those small microspheres, we can really use them as a handle to actually trap biomolecules, exert forces, and perform single molecule experiments. To give an idea about the impact that optical tweezers had in science, just last year, Arthur Ashkin, which was one of the pioneers in the development of the optical tweezer technology, was awarded a Nobel Prize for the invention of optical tweezers. But how does it really work? So how can light really trap small objects? So let's, for one moment, imagine to be a photon and hit upon a small glass sphere. What happens? When the photon hits small glass sphere, what happens is this photon is being refracted, so it changes its direction upon the entering of this small sphere. And once it exits, even it's further deflected. So that means that a change of momentum occurred in the photon from the beginning to the from before or after we hit the small sphere. So a change of momentum, of course, momentum has to be conserved, as we know from basic physics. That means that the same momentum change that the photon experienced are equal but opposite direction, momentum change was act on the bead. So actually the deflection of light applied a small force on that bead. So what happens when instead of having a single photon, we have instead a lot of photons, all of them interacting with a single bead. And if we think that these photons are converging into a light focus, each of these photons will slightly change momentum. And that means there will be a net momentum or force that will direct the sphere towards the focus of the laser. And so this is the basics of optical trapping. The second technique we combine is single molecule fluorescence using confocal microscopy, where again, we use a convergent beam with a focus, and then we record the light that is emitted from this focal point, and we project it into single photon detector. By moving this point across our sample, we can build images, and that allows us to really see fluorescence images, as you see the one below. Because of the sensitivity of the detection system and of our device, we are so sensitive that we can detect individual fluorescent molecules. And how can we be sure that we're looking at individual fluorescent molecules? Because when we observe the fluorescence particle in time, after a certain amount of frames, this molecule photobleaches, which means that it abruptly goes from emitting to a not emitting state, which is really the fingerprint that we were looking at an individual single molecule. There is a third important ingredient in performing a single molecule experiment, which is the laminar flow microfluidics, which is a method where that allows us to flow all individual reagents of the sample together into a sample chamber and construct the single molecule assay directly in situ. And that really enables us to, in a fast way, efficient way, perform many experiments one after each other in a high throughput way. And I will now run a few movies that will give you a more direct visual of how this microfluidic system really works.
We start at our microfluidic system. Here we insert beads, DNA, and proteins of interest in up to 6 fluidic channels. Pressure drives the flow to the microfluidic chip. The laminar flow environment ensures that no physical barriers separate the flows, allowing you to freely navigate between them without any risk of mixing. After our fluidics have been connected, we're ready to switch to our workstation.
So this is really the first step. So as a user, what you do really is just load your sample onto the syringes, and there is a pressure-driven flow that injects those reagents into a microfabricated glass chip. What happens once the fluids enter this glass chip, you will see in the following movie. As we said, we inject the pressure liquids inside our microchip via pressure-driven flow. And this microfluidic chip has multiple inlet channels. And because of the laminar flow condition, each of these reagents flow next to each other without mixing, even in the absence of physical barriers. That allows us, for example, to just activate the trap inside the channel where the beads are floating. That allows us to pick the 2 individual beads, and that allows us to move from the beads channel into, for example, the DNA channel. And in the DNA channel, what we can do is use those beads to capture individual DNA molecule. And we can be sure, for example, we're having an individual DNA molecule, because by moving the beads with respect to each other, we can apply a controlled tension on the DNA, and by recording the tension and measuring the tension, then we can really be sure that we're looking at an individual molecule. Once we know that we have an individual molecule, then we can start our experiment, and we can incubate this DNA molecule in multiple protein channels in a sequential manner. And once we've done it, then we are ready to image the product of our biomolecular reaction, and then we switch on our confocal microscope, and then we scan along our DNA segment. Because confocal scanning, because we have a single one-dimensional system, a single DNA molecule, that allows us to go very fast, and then we can scan along the DNA, and that gives us the information about the position, the intensity, and the distribution of each of the proteins which are bound onto the DNA molecule. And then we can record at the same time the fluorescence signal and the force signal at the same time. So this is really like one of the key technological advances of Lumicks, the ability to be able to correlate in a single hardware, in a single software, high-resolution force detection and high-resolution fluorescence detection. So Lumicks, again, a small historical background, started in 2014 as a spin-off from the lab of Gijs Welten and Erwin Peterman in Amsterdam. The lab has a strong expertise in DNA-protein interaction, and that was our initial focus of our company. DNA-protein interaction is a very large field of application, and in a typical DNA-protein interaction assay, a single DNA molecule is extended, and we can obtain a lot of information about different aspects of genome metabolism. We can say DNA repair, DNA organization, DNA replication, transcription, or even translation. In general, what we observe is we look at individual protein moving or interacting with each other or with a single DNA molecule, and then we project it into this form of chymograph type analysis, where on the x-axis you have time, and on the y-axis, you have a line scan of the fluorescence along the DNA molecule, and then we see, labeled in different colors, different individual proteins interacting with each other and moving along the DNA. Because of the single molecule sensitivity, we can also be very quantitative in this measurement, and we can determine if the interacting proteins are monomer, dimers, trimers, or any other form. So by looking at the fluorescence intensity, because we have single molecule fluorescence resolution, then we can also determine the actual size of these protein complexes on DNA. Also, I want to highlight a recent example from literature by one of our customers, David Rueda at Imperial College London, that together with the company AstraZeneca, they identified a novel mechanism where Cas9 is rendered insensitive to the target by force. And that is important because actually in many cellular processes, as they describe in the article, the DNA structure is under stress during transcription or DNA replication. And those stressful genetic conditions can expose those off-target sites to Cas9. And that is actually one of the underlying cause of the observed Cas9 off-target activity. And I want to just show you a movie about how this actually DNA stretching really induces Cas9 off-target activity. So what you will see in this movie is a single DNA molecule trapped by two individual optical tweezers, and we will move it, we will extend it gradually, and then we'll observe as a reaction of the extension how much Cas9 will bind on a single DNA molecule. And, oh, I'm sorry. So you see at the low force, there is no binding of Cas9, and we start increasing the force, we start observing Cas9 binding in an unspecific way across all the DNA molecule. And when we further relax the DNA, then we can see that this binding is and unbinding are reversible. If you want to know more about Cas9 off-target effect, please, this is the reference of the article we just published a couple of months ago in Nature Structural Molecular Biology. Okay, as a scientist, so what do we need to perform these experiments is really the ability to stretch DNA molecule, to image DNA molecule, but there are other experiments where, for example, there are different requirements that are needed. For example, to study protein unfolding. And for protein unfolding, it's very important to have ultra-stable, fast force detection, control the temperature, and even automate the experiment. And that's what we introduced about last year with the introduction of the C-Trap G2 system. And I'm going to go a little bit now into detail about what protein unfolding experiment look like. The assay is very similar to what you've just seen a moment ago on a DNA level, but this time we can pull on a single protein. And by applying force on a single protein, we can see when unfolding or a specific domain occurs, or we can see when a specific conformational change occurs. So what we do is we can site-specific label the protein and a single amino acid, attach small DNA linker, and we can start and apply the force. This is an example of this experiment where we unfold a single protein. And in this case, it's the protein calmodulin. And what we see as we extend the DNA molecule on the x-axis with the protein, the force starts to ramp up. And then at a certain point, we observe a specific rupture event as a drop in the force. And that corresponds to the force required to unfold the protein and go from a three-dimensional folded structure to a completely unstructured polypeptide, which is about, in this case, 8 piconewton. Protein have a very interesting feature when they are subject to force. Is in some cases, this folding and unfolding behavior are actually reversible. And we can see that by looking at the force switching between the two force states in an alternating manner. So when the force is in the high state, it means that the protein is folded, and when the force is in the low state, it means the protein is unfolded. And we can see that this protein can cycle between the folded and unfolded state multiple times in a reversible manner. So we can really study the kinetics and the dynamics of this folding unfolding process at the single molecule level. What we can do with this information, with this data, scientists are now able to reconstruct exactly the free energy landscape from this force spectroscopy measurement. And then we really are able to, from force spectroscopy measurement, come up with an underlying free energy landscape that regulates the three-dimensional stability and structure of the protein. Also, to give you an outlook, actually those type of experiments can also be equally done with the RNA molecule, and where we use a single RNA structure, RNA hairpin, or other more complex structure. We link that to double-stranded DNA duplexes, and then we can perform the exactly same experiment. And you see here on the right, again, another force extension trace where we see an RNA unfolding, again, happening at the force between 6 to 9 picon. So so far, I've only discussed about the use of optical tweezers and confocal microscopy, but I want to mention that there are many other imaging possibilities. First of all, the confocal microscopy could be expanded to perform super resolution measurement using STED, super resolution STED. But also, we have just released an entirely new suite of imaging modalities that we call the surface assay toolkit, because those are imaging modalities which are most important when we're looking at samples in a proximity to a glass surface. And those are really 3 imaging modes that can be integrated in a fully modular and compatible manner with both Lumicks optical tweezer system. And I will go into detail right now. So one is called IRM, which is a fast 2D imaging methods, which allows us to see biomolecules in a label-free fashion. And the 2 other technologies are wide field and third fluorescence imaging. Wide field allows us rapid 2D imaging deep in solution. For example, when we have trapped a single DNA molecule far away from the glass surface, then we can observe it at higher temporal resolution. And third is very important when we have a sample that are sitting very close to the surface, and then we want to observe those samples in a highly sensitive way while being able to have efficient background rejection. So interference reflection microscopy is a technology that we've just introduced this year. From a short historical perspective, this technology was for the first time pioneered long time ago in 1964, was proposed by Curtis, but only until 2018, the 2 different research groups, one in Yale and one in Germany, were able to bring this technology to a performance level where it could enable visualization of single molecules. So Simmert et al. And Mamad et al. From the labs of Eric Schaefer in Tübingen and Joe Howard et al. Recently demonstrated that individual microtubules can be detected. So the implementation of technology is quite straightforward, because we use a LED light source that is coupled into our objective, and we detect the backscatter light from the sample, and we perform an interference measurement between the backscatter light from the sample, for example, a microtubule, or from the glass surface. And that via a tube lens, we project them into a camera. So what did we choose this to develop an IRM module? So it's a very sensitive imaging technique. And you can see in the image on the right that microtubules can be easily detected in a dynamic way with a high contrast. And a very important criteria is also that IRM can offer a very important advantage when integrating with optical tweezer, for example, with DIC, where the presence of a Normansky prism typically limits the integration with optical tweezer. So this is a thing very, very interesting, because we can now see individual microtubule on a surface without the need of fluorescence label. And we can do that even with the high sensitivity. So this is give you a quick idea of the workflow that we employ to see microtubules with high signal to noise ratio. First, we take a raw image in as short as 2 milliseconds. We look at a background image to make sure that we are not looking at any potential artifacts to dust or dirt on the glass chip. Then we have a background subtracted image, and then we can further average, and then that allows us to really improve our signal to noise ratio to above 18. So now we go a little bit more into biological application. In this movie, we show one of the most intriguing maybe aspects of microtubules, which is the ability of assembling and disassembling over many cycles. This is actually called dynamic instability, which is a behavior in which microtubule, which has been observed for a long time, which is an interesting behavior where periods of growth of microtubule is interrupted by abrupt shrinkage, and those are called microtubule catastrophe. And from this movie, which lasts for over 20 minutes, you can see an example of how IRM can detect this dynamic instability. And now this data is very quantitative and very clean and allows us to really obtain kinetic information about dynamic microtubules. A very interesting opportunity when we have the ability to measure both fluorescence and IRM signal is to actually use the two technologies simultaneously. So by having a system with two cameras, one dedicated for the IRM and one dedicated for the fluorescence, and then we can easily combine the two imaging solution and have a correlated wide field or turf with the IRM. And this is very important. I want to mention that this really also allows to, for example, study more complex biochemical reaction once we have the ability to do multicolor fluorescence in combination with level 3 imaging. And then we can do in a multicolor fashion. So we can use 488 nanometer laser, 532 or 561, and a 638 nanometer laser, which is in the red band. The second principle that also guided the development of the surface assay toolkit for studying protein interaction with microtubule on a glass is the development of the hardware module, but also is the actual workflow that allows the user to go from manipulating single visual imaging to actually perform sensitive measurement and observe them for a long period of time. And those are the three important criteria, because we should be able to simultaneously measure forces while doing imaging. We should have the measurement sensitivity to measure the events as small as possible. And we should also have the stability such that rare events are being detected. So those are the three criteria that really guided this technological development. Here you see an example where you will see an application of the optical tweezers combined with the IRM, where a single optical tweezers is trapped in a single bead, and a single kinesin molecule is brought in proximity to a microtubule close to a surface, and then we observe the processive motion of the kinesin in real time. And you can see the fast stepping of kinesin performing roughly 8 nanometer step at in a highly processive manner. And we can also do that from a force clamping mode, where, for example, we keep the force constant and we follow the displacement in time of the motor, but we can also do in a distance clamp mode, where we are actually looking at the stalling force of the motor. What you see in this graph, as we start the experiment, the kinesin starts to walk, and then reaches at one point it reaches the force of 1.5 piconewton, and then it stalls and releases from the microtubule and comes back to its original position until it goes for a new run again. So in general, just to give you again an overview, I think what I want to show today was that combining optical tweezers, single molecule fluorescence, and microfluidics, then we have access to a broad range of biological application: DNA, RNA, protein interaction, studying protein unfolding and conformational changes, but also the activity of molecular motors, microtubules that are important element of cellular structure and transport in our cell. I want to quickly highlight a few other applications that are really coming up from our users. One, before closing off the talk, one is about studying membrane protein interaction, and specifically dynamic kinetics and sensitivity of membrane fusion. This is a work published in 2015 in Nature Communication by our founder, Guy Raute, where we use optical tweezers together with fluorescence microscopy, and where individual molecules of transmembrane protein called DOCK2B was studied in the presence of fluorescent phospholipids. And the idea here is that while bringing the two Bs together allows a controlled way to trigger membrane fusion and membrane fission reaction. And when the two Bs are brought together and moved apart, then we can see the formation of those tubes in the bottom figure. And those tubes are really the result of a successful fission of the two initially separated lipid bilayer. And what we notice is only in the presence of the specific protein, DOCK2B, at the concentration of 0.74 micromolar, the interaction was strong enough such that even that could stand the force of like 50 to 100 piconewtons, so showing it was a very stable biological structure, and that DOCK2B was really essential for the formation of this intermediate. Another upcoming field of study is the study of liquid droplets, which are biological compartments that are made by protein inside the cell, where protein phase separate with respect to the environment to create sort of a small membraneless compartment where biochemical reaction can occur at a highly enhanced rate. This was studied in the laboratory of Tony Hyman at Max Planck Institute in Dresden, and in the context of a ALS-associated protein, FUS. ALS is a neurodegenerative disorder, and an undergoing theory is that the loss of this liquid-like behavior of this protein droplet was associated with the onset of this disease due to a mutation. And so they used optical tweezer with fluorescence microscopy to study actually how genetic mutations or aging affect this liquid-like behavior of this droplet. So the experiment was performed as such. Two optical tweezers were used, and an individual protein droplet was manipulated, and then mechanical measurements were performed. Unfortunately, I cannot play the movie for this time, but please, I refer to this publication of Physical Review Letters in 2018. The last application I want to show is about mechanobiology, where optical tweezers are combined together with live cell microscopy. And that's something like a very interesting finding, that actually biochemical signal can be modulated by force. So live cell actually can sense the surrounding environment, and with optical tweezer we have a very, very sensitive and controlled way to apply specific forces to specific parts of the cell, and we can measure the force response of the cell, but we can also visualize the activation, the signaling of the cell, and the specific is a calcium flux is enhanced once we apply force on the cell, as you see the large increase in the red signal. Okay, so maybe to wrap up, so I gave you an overview of the most interesting application of the C-Trap. Before we close off, I'll give you a run through to what the C-Trap really is. So it's an integrated single molecule analysis platform that combines dual up to quadruple optical trapping methods. It is designed for high resolution and high stability measurement. It contains a 3-color scanning confocal fluorescence microscope, but can also be integrated with a wide field and turf and IRM system, as I discussed. We show single molecule and single photon sensitivity with the capability of upgrading to super resolution STED. It's entirely automated. So as a user, you just sit behind a computer and you have the ability to script entire experiment from your Python notebook. And the microfluidics really enables high throughput and efficient experimentation because of the in situ assembly of the single molecule assay. So with this, I would like to thank you for your time.